Welcome to the first video in our Viruses and Prions series. This video is going to be short and sweet, and we're just going to talk about general characteristics of viruses. Now, the first thing you have to understand is that viruses are not a living organism. And we're going to talk about all the reasons why that is on the next slide. But a lot of people are surprised by this because in normal societal non-sciency conversations, you hear people saying things like, you can use this to kill a virus. Well, technically you can't kill something if it's not alive, right? So first and foremost, viruses are not a living thing. Therefore, we shouldn't call it an organism, okay? What viruses are they are obligate intracellular parasites. When we say obligate, that means they are 100% dependent upon a host to do anything. So if you had a virus particle sitting on your laptop, it wouldn't be able to do anything. It would not be doing metabolism. It would not be making energy. It would not be replicating. It would do nothing because a laptop isn't alive. Now, if you breathe in a virus, it may be able to then take over your cells and start making you sick. Okay, but it is completely dependent upon a human. Oh, I'm sorry, not a human. 100% dependent upon a host. It's intracellular because it has to get within actual cells. That's what intracellular means, within cells. And because it is intracellular, once it gets in there, it's going to basically take over the cell. It hijacks all of the cell machinery. It uses cell ribosomes, polymerases, ATP, nucleotides, all the things the cell normally uses to build macromolecules for itself, the virus takes over. And it's a parasite because the host is harmed while the virus technically benefits. Now, viruses are really, really small, much smaller than bacteria. They typically range in size from about 20 nanometers to 1,000 nanometers. And if you look at this picture here, you can kind of get an idea of how small things are. So here's a frog egg at about a millimeter, and you don't need to memorize these examples or sizes. It's just to make a point. And then you can see how much smaller animal and plant cells, some eukaryotic cells, are. Zooming in, some bacteria are about the size of the mitochondria. Remember our conversation on the endosymbiotic theory? That may not be too surprising. And then look, now we start getting into viruses. Okay, so some viruses like smallpox, they're a bit smaller than bacteria, but then you can get down to some others like polio, and they get pretty tiny, okay? And then smaller than them are going to be some of the macromolecules like lipids, proteins, things like that. And then, of course, the smallest on this picture is the atom. Okay, so viruses are really tiny. Another thing is viruses have a host range. Basically, that's the range of organisms that a virus can infect. Some viruses can only infect bacteria. Some can only infect plant cells or animal cells. We can get even more specific. Some viruses can only infect humans. Maybe it can only infect, pigs or, other non-human animals. Every virus is different and it has its own host range. And we'll talk more about what dictates that host range in just a minute. And then the last point I really want to drive home right now is that not all viruses are alike. We are going to look at several different viruses, and you're going to see that structurally and genetically, they are very, very different. Now, what do all viruses have in common? All viruses have nucleic acid, so DNA or RNA, and a protein coat that's going to surround and protect that nucleic acid. Okay, so at the simplest level, all viruses have at least a nucleic acid and a protein coat surrounding it. Now let's talk a little bit more about why viruses are not considered living. First of all, they are acellular. That means they are not made up of cells. And according to the cell theory, all living things are made up of cells. So automatically, by default, viruses cannot be living because they're not made up of cells. They don't have a plasma membrane. They don't have mitochondria. They don't have ribosomes. They don't have cytoplasm or cytosol, okay? They don't have DNA polymerase, RNA polymerase, transfer RNAs. You get my gist. They don't have any of that. So again, that's why they're obligate and intracellular, because they have to get into our cells to take over in order to do anything. Another reason viruses are not considered living is because they don't contain both DNA and RNA. A virus comes equipped with just one or the other. They may have DNA, but that's it. They may have RNA, but that's it. Living things have both DNA and RNA because remember, transcription is going on inside of living things. As I've already mentioned, viruses cannot replicate on their own. They cannot do asexual reproduction. So again, unless they're in a living host, if they're not in a living host, they can't do anything, okay? They cannot maintain homeostasis. There's no way of like regulating anything based on the environment. They don't respond to stimuli. If it gets too hot, gets too cold, they can't do anything, okay? And they don't produce any energy or do any type of metabolism on their own. And these are all things on this table that living organisms can do. Okay, so there's a lot of boxes that viruses do not check in order to be considered alive. Now, let's look at viral host range. So I mentioned that the host range is the range of organisms that a virus is able to infect. Okay, so some viruses can only infect one species, maybe two. Okay, when it's very limited on what it can infect, we say it has a narrow host range. There are other viruses that can infect all sorts of different animals, for instance. We say they have a very broad host range. So what determines if it's a narrow or a broad host range? What determines what a virus can successfully infect? Well, that's going to be the spike proteins. Okay, that's going to be these proteins that are sticking out of the virus. Okay, we're going to talk a lot more about these terms that you see on this picture in the next video. But these spike proteins, right, they are the outwardmost structure. They are going to be able to bind to specific receptors on the host cell. Now, the receptors on the host cell are not meant for the virus, okay? The host does not want to get infected by this virus. Rather, these viruses are being sneaky. Their spike proteins resemble the shape of something the host does want to let in. And so it tricks the host cell. When the virus binds to that host cell receptor, the host thinks it's getting something that it wants. And so it lets the virus in, and then it regrets it, okay? So again, we're paying attention to the shape of these spike proteins. In this example, they're round, okay? And that's gonna be important. So let's look at potential host cell number 1. Notice the receptors on its surface are square. So imagine if this if this spike protein tried to bind to this host receptor, it wouldn't fit, right? It would just kind of bounce right out. So potential host number 1 is not going to work because the shape of the receptor is not complementary, not similar enough to the shape of this capsid protein. I'm sorry, it's the spike protein. Sometimes the spike proteins are capsid proteins, but in this case, the spike is much more prominent. All right, so let's look at potential host cell number 2. So the receptors on this host cell are triangular. So what's going to happen if this circular spike protein tries to bind and kind of stick to this receptor? It's not really going to work, right? Because the shapes aren't the same. Because remember, y'all, in the body, in reality, things are constantly in motion, right? And so if the shape of the spike protein is not very, very similar to the shape of the receptor, as things move about, they're going to fall, they're going to separate, okay? So these shapes are not similar enough for the spike protein to attach to that receptor. So let's look at potential host cell number 3. Now this one is round, okay? So this is what the virus is looking for, because when this spike protein comes into contact with this host cell receptor, it's much more of a lock and key fit. So they're going to be similar shape. The host receptor is going to be like, oh, hey, yep, I'm looking for you. You got the right shape. Come on in. And then, like I said, eventually the host cell will regret that decision. So in this example, potential host cell number 3 would be an organism that the virus can infect. Okay, so to determine the host range, the host has to have receptors that are complementary in shape. It's a good science word to remember, complementary in shape to this viral spike protein. All right, y'all, that's it for this video. Let me know if you have any questions.